What's up? I welcome you all to this conceptually connected series for science and technology for UPSC on the Platonic YouTube channel. Myself Himanshu, and today is topic 4, DNA and mRNA vaccines. Guys, we have already completed topic 1, 2 and 3 which are available on the Platonic YouTube playlist for the science and technology. Additionally, we are also running several other series such as environment conceptually connected series and other strategy sessions which you can go and visit the channel and get the benefit. And guys, share as much as possible. Let's begin today's topic. So as you know that today's topic is about DNA and RNA, mRNA vaccines. But before that, we need to understand that what are the two types of nucleic acid. So as we know that basically there are two types of nucleic acid. One is the DNA, other one is the RNA. So DNA and RNA, as you can see the structure, this is DNA stands for deoxyribonucleic acid and RNA stands for ribonucleic acid. So there is a basic difference, which is that the DNA is double stranded, whereas the RNA is single stranded. You can see from the diagram itself. But there are several other differences also which we will see, which will be beneficial for some additional basic questions if it is asked in the examination, such as that the DNA contains the deoxyribose sugar, whereas the RNA contains the ribose sugar. Instead of thymine, the nitrogenous bases, uracil is present in case of RNA instead of thymine, whereas in case of DNA, uracil is not present. We have already seen that DNA is double stranded and RNA is single stranded. So additionally, one more important point to notice is that DNA is more stable compared to RNA because there is no free hydroxyl group present in DNA, whereas it is present in the case of RNA, whereby it is hydrolyzed easily by the alkalis. And because of that, the lifetime of DNA is comparatively high, whereas in case of RNA, it is short-lived. And because of this nature in case of RNA, because it has the free hydroxyl group, so it becomes catalytic in nature. And DNA is no more catalytic because it doesn't have the free hydroxyl group available. And we have already seen the structure that DNA is double stranded and RNA is single stranded. Both the strand in the case of DNA and the RNA, like in that case of DNA, the strands are anti-parallel in nature, okay, whereas it is not the case in case of RNA. And these are the four nitrogenous bases, basically cytosine, guanine, adenine, and thymine. So instead of T, U is present in case of RNA. So if any basic questions comes about DNA and RNA, so probably you can use this piece of information to address those questions. So guys, let's move forward. So this mRNA and DNA vaccines are a hot topic because, as you know, that during the COVID pandemic, several vaccines were designed and several other vaccines were made to cope up with this global pandemic, which was COVID. So vaccines, the basic facts about vaccines is that the vaccines, they prevent or mitigate the infection by providing protective immune response. So how the vaccines are, what are the different type of vaccines are? So basically they provide immunity. If you inject vaccine into an individual, so his body will develop an immunity against that particular kind of virus against which this vaccine was isolated or this vaccine was made. So the individual will be saved by the vaccine. So there are some differences between a drug and a vaccine, which we'll be seeing in the upcoming lectures. But as of now, we'll restrict ourselves to vaccines. So as you know, there are different types of vaccines. There are live vaccines, which are taken from its original biological resource. Let us say live form of viruses, live form of bacteria, live form of viruses are generally taken and they are converted into a vaccine and they are utilized directly. Then there are inactivated vaccines. So this is the second level where the virus particles are inactivated and the vaccines are made. Then there are viral vector vaccine. They are replicating and non-replicating type. Then there are DNA vaccine, mRNA vaccine, and subunit vaccines. So DNA vaccines and mRNA vaccines will be our will be studying today. DNA and mRNA vaccine in particular. Subunit vaccines and other types of vaccine will be seeing in upcoming lectures. So what are the most important characteristics of the vaccines? So the most important characteristic of the vaccines are they're almost always taken from the biological product. As I told you that in case of live attenuated and inactivated vaccines, virus particles are taken, viruses are taken, and they are converted into vaccine, like the cowpox vaccine as it was discovered, like in the case of smallpox vaccine. So the activated, attenuated, or inactivated vaccines are basically used. So as you can see, they are having biological origin. Then mostly they require cold storage because storage they require special condition of storage. Then large molecules are usually administered parentally, and then they are supposed to protect whole population, because what happens is that population develop herd immunity, and this herd immunity was additional topic which is very important. We'll be seeing in the upcoming lecture. What happens is that let us say this is population A and this is population B. Now if population A, within population A there are various individuals, and these two populations, subpopulations, are living in one particular geographical location, let us say X. Now if population A has been vaccinated fully, so the individuals among these A set of subpopulation will develop an herd immunity. So probably even if the infection arises again, even if the virus mutates, even if the virus again attacks, so because the population, the subpopulation is already getting immunized because of the vaccine, so they will develop herd immunity. So examples of vaccines in news were Covaxin, then there was Sputnik 5. So we'll start and we'll see that how mRNA vaccine works. So let's begin with the mRNA vaccine. Now mRNA, as you know, mRNA has a characteristic that mRNA is directly converted into protein. So mRNA are directly converted into protein, and this is the basis for the mRNA vaccines. So let us say that scientists they designed the mRNA based vaccines upon the coronavirus. So what do they did actually was they generated the mRNA sequence for the virus spike protein. So this was the mRNA sequence which was made, and then they took the mRNA sequence and they used the lipid coating. So because mRNA needs to be delivered, so they encapsulated this mRNA into a lipid uh, vehicle for delivery, and then this was converted into an mRNA vaccine. Now, the RNA sequence, which was taken from the spike protein, is a blueprint for making the spike. Uh, so it is injected into the individual, and once it arrives, what happens is the mRNA produces millions of copies. It gets replicated, and millions of copies of the spike protein are made inside the cell. Now, what happens? As the protein fragments are made, the immune system starts to produce antibodies which are specific against the spike protein, which are specific against the spike protein. So those antibodies are made, and this is how the immunity is achieved by the mRNA based vaccine. So this is how Pfizer basically designed this coronavirus specific vaccine, and this was mRNA based vaccine. So guys, let's move to the next phase. Now, in case of DNA based vaccine, so we have seen how mRNA based vaccines have been designed. So in the case of DNA based vaccines, so how the DNA is being made. So DNA based vaccines are made. So as you know, the central dogma is that from the DNA, mRNA is synthesized, and from the mRNA, protein is synthesized. So this is the central dogma. This is the central dogma of molecular biology. So let us say in the case of DNA based vaccine, this time the virus we are taking into consideration is the West Nile virus. This West Nile virus is also in news. So what we do, we destroy the virus, we convert we isolate the RNA initially, which is single-stranded genetic piece of information. So we take RNA. So we take RNA, and this RNA is then converted into DNA. RNA is converted into DNA. So reverse transcription is done. So RNA is converted into DNA. So RNA is converted into DNA, and then what is happening? That particular, let us say there is particular protein of interest. Let us say in this case M protein. So what we take, we take the gene of interest which codes for the M protein, and then the same and RNA is converted into DNA via reverse transcription, and the DNA sequences are synthesized in the synthesizer, and based on the DNA sequence we make short palindromic sequences which are known as primers, which are specific to the PRM gene, which is the gene of interest, and from these primers we generate the cDNA. So uh, from the DNA we make the cDNA, which is complementary DNA. So this is little bit more technical, but once the cDNA is made, so then we use plasmids. So as you know that plasmids are autonomous uh, replicating sequences which are found in the bacterial systems, and they have copy number. So depending on species to species of bacteria, basically we use E. coli initially. So E. coli is and this is extra chromosomal in nature. This plasmid is extra chromosomal in nature. So it provides certain properties. Plasmid we'll see in separate lecture that how and why plasmid is important. But as of now, we'll see that when the cDNA is integrated in the plasmid, then plasmid is grown, it is multiplied, and expression, basically we use expression vector over here. So expression vector is used. So there are several types of vectors. So we'll see in additionally, so there are types of vectors also. So there are expression vectors, then there are cloning vectors, there are shuttle vectors. So there are several kinds of vectors, but in the cloning vector, expression is also there. So finally, this gene will be expressed, this PRM and this E gene, it will be scaled up in the lab, and then the protein will be made. So protein purification is done by chromatography, which is one of the methods, although this part is of downstream processing, and there are several other ways of purification of the protein. But these purified plasmids are basically the genes which make up the vaccine. So as you can see, compared to the mRNA based vaccine, DNA vaccines are little more tedious to manufacture, and it is also a very lengthy process. So let's come back to the topic. So what are the overall advantages of the vaccines? So as we know that vaccines are more cost effective and they are produced quickly uh, when compared to the traditional vaccine. Now, as you know, with every upcoming or new technology, there are several challenges associated, e- uh, even when there are some small scale improvements. So basically, this whole game is of trial and error. So a lot of experiments are conducted before the final clinical trials are cleared for those particular DNA or mRNA based vaccine. Then, of course, skilled labor is required, high overall capital if we see it is required. Then there are some policy bottlenecks also, and especially ethical, legal, and social issues they arise. So a lot of protest and agitation is also there sometimes, because the gene of interest is taken from what particular source or animal becomes important as well, and how much it is safe for human trials. Then more validation and cross validation of the clinical trials needs to be done. And one more thing is that if there are some zoonotic diseases which are new in nature, so it is then difficult to design mRNA or DNA based vaccines very quickly. It is more easier to design mRNA based vaccine compared to DNA based vaccines. So let us see that what are the differences between mRNA and DNA based vaccines. So as you know that RNA vaccines or the mRNA vaccines, they are made from the mRNA itself, okay, and this DNA vaccine is made from the piece of DNA. So they are then how they are delivered. So they are delivered using liquid nanoparticles, whereas DNA based vaccines are delivered using plasmids. So what is happening is that mRNA makes the protein, and protein basically illustrates the antibody formation. Whereas in the case of DNA based vaccine, it is much slower to make antigen because first the DNA enters the nucleus. So DNA will complete that entire cycle. So DNA will be there, so mRNA will be made, and then protein will be made. So one step longer is there. That is why a slower antibody formation is there. And in this case, directly from the mRNA, protein is made. So it is more quicker compared to DNA. But there is one advantage in case of DNA based vaccines, that they are very highly stable compared to mRNA based vaccines, which are low level of stability is there. Then let us come to the advantages. So as you know, as we have already seen, that RNA based vaccines are protein synthesis ready, okay, and they are less riskier than DNA based vaccine for the obvious reasons which we have discussed before. And the overall process of making antibodies is faster, both in the case of mRNA vaccine and DNA vaccine, more faster in case of mRNA vaccine compared to DNA vaccine. But there are several disadvantages, that only a part of the mRNA is used. So there might be situations that the immune response is not very strong, and therefore we require booster doses. And additionally, DNA vaccines are more time taken compared to the mRNA based vaccine. So all these things are there which we need to understand when we are studying or we are observing mRNA and DNA based vaccines. Now, what is the way forward in this whole uh, scenario of mRNA and DNA based vaccine, especially after the COVID pandemic? mRNA vaccines are uh, in synthesized in huge number, and they have been very successful with a high efficacy percentage. So first of all, we require a robust ecosystem that facilitates cutting edge research, because that is where the new innovations arise. And then there are establishment for state of the art manufacturing facility. So state of the art manufacturing facility is required, and because of which there should be careful and scientific data validation and clinical trials. Okay, and we need to address ethical, legal, and social issues, if any, if anything is there. Then there is 5I approach, which is although it is applicable at various other places as well, we can use over here, which is inclusiveness, indigenization, innovation, investment in infrastructure, and international cooperation. So with all these things, we can speed up the process. We can speed up the process of making the vaccines commercially viable for a large set of population, both in case of developing as well as developed countries. So thank you, sir. Thank you all. This was the topic about mRNA and DNA-based vaccine. I hope the lecture is useful. See you in the upcoming lectures. Thank you for being Platonic. Goodbye.